Okay. Do you want me to start? Do you hear me? Hello. Do you hear me?
I can hear you.
Yeah. I know. I'm wondering whether like the people in like Dubai hear me. I don't hear Rashid anymore. Um, hello. Yes. Yeah. Do you want me to start? Okay. Okay, so thank you very much. It's really a pleasure to be somehow with you on Zoom. I will be talking today about solving biology problems using artificial intelligence, and I'll be mostly talking about AlphaFold2 for protein structure prediction and our own AI system for predicting proteins. So very briefly, proteins are a remarkable nanomachine. Pretty much everything all functions in any living organism is achieved by a protein or complexes of proteins, and their function to a large extent is embedded into their exquisite three-dimensional structures. Here we have few proteins and how they interact. So to understand life at the microscopic level and the mechanistic principle underpinning
Recording in
life,
progress.
it starts with understanding proteins and their structure. On the other hand, many diseases are related to situations where proteins are misfolded. They don't get the right shape. So what we see here, these are protein aggregate in orange surrounding neuronal cells, and they are causing neurodegenerative diseases such as Alzheimer's disease. And here, finally, what we see, this is an interaction between a small chemical compound, a drug, in a protein. And often when you want to design a drug, you need to target a specific pocket in a protein, we call it the active site. And this also starts with understanding the structure of a protein. So to sum up, to understand life at the mechanistic level, to understand many diseases, and to start thinking about drug discovery at the molecular level, we need to understand proteins, their structure, and next, their function. So the question that I would like to raise today, what is the bottleneck? What are the limits today that we are facing for having such an understanding, both for the principles of life, the diseases, and therapeutic approaches? The main bottleneck is experimental data. So what I'm plotting here in blue is the growth of available data in sequence space. And then as we see, there is an exponential growth since the Human Genome Project. And the reason for that is quite simple to understand. Sequencing today is rather cheap and it's broadly available. So currently we have about hundreds of millions of sequences available in databases such as UniProt. On the other hand, if you look at the data available in terms of protein structures, this is the orange line in terms of like a growth per year. And despite the fact that we have some growth, but this is very modest, it's only linear, and we don't expect anytime soon to bridge the gap between the orange point, the number of available structure in public databases, and the blue histograms, which again reflect the number of sequence. And the reason is quite easy to understand. Resolving experimentally protein structure is very expensive. So typically, if you want to refine the structure of an individual protein, you need to spend hundreds of thousands of dollars. It is tedious. It requires the work of PhD students and postdocs, and it takes a year. So there is no hope, at least in the near future, to bridge the gap between our understanding at the sequence level and the structural understanding. So therefore, the question, if experiments are not the path for bridging the gap, what could be the alternative? How can we acquire this structural knowledge? The starting point is a statement, observation, vision made by Enfinson in the 60s, which is that pretty much all the information needed for folding a protein, so this is what you see on the right, is a folded protein, is encoded into the sequence of amino acids. And if we accept this premise, the idea now is to find a mathematical map from the sequences of amino acids to the structure, given the fact that we have a large amount of data in the sequence databases. So since Alfinson, this became one of biology's major challenge to find this map from sequence to structure. And in the last year or so, there was a major development in the field, mostly thanks to the combination of co-evolutionary idea from biology and artificial intelligence. This major milestone was achieved by Deepmind. This is the premier AI company owned by Google. And to give you a sense about their achievement, here I'm plotting in green an experimental structure. So this is what experimentalists measure. And in blue, these are the predicted structures from the Deepmind system called AlphaFold 2, which will be the main topic of my talk today. And as I hope you realize, there is a very good match between the green structure and the blue structure, suggesting that in many cases, now we have an AI system, AlphaFold, that is capable to predict structures starting from sequence with very high accuracy, an accuracy that is now of interest to experimental biologists. And this is really a turning point, I believe, and many of my colleagues believe, in structural biology and biology, moreover. Just to give you a sense about this achievement, I'm plotting here in the y-axis the accuracy of our predictions, how good are our model for predicting structure, and in the x-axis, the time evolution in the last couple of years. So if we look back 10 years ago, around 2012, that was the situation, this point. Our best predicted structures were above 10 RMSD, which is a metric for measuring how good is your protein. And those results were not of interest to biologists, because with this resolution, you cannot say anything about the biology and the mechanistic underpinning of those structures. And what happened, starting mostly in 2018 with AI and AlphaFold, is you see that we have a substantial drop, which means higher accuracy. And this is the situation today. We are around 2 angstrom on average for most protein. And this is a very exciting situation for computational biologists, AI experts, but also experimental biologists. The work of AlphaFold on protein structure prediction was recognized by Science, again, premier scientific magazine, as the breakthrough of the year for 2021. So this is, again, as I said, a turning point in the way how we are practicing structural biology. And I hope at some point, biology, moreover, disease treatment and drug discovery. So the plan of my talk today is, first, I want to give you a sense, an intuition. How did we reach this milestone? Why AlphaFold is so powerful as an AI system? And I will turn to board games and AlphaGo, another important AI system that inspired AlphaFold. On the other hand, I will give you a glimpse about AlphaFold 2. What is this system? How it works? And if I have time, I will talk about an alternative AI system that we have introduced with my group that is based on single sequence prediction. And we believe that our own system can tackle questions and problems that are currently beyond AlphaFold 2 system. So from AlphaGo to AlphaFold. So as I said, the major milestone that I mentioned, which is starting to predict protein structure with very high accuracy, was achieved by Google Deepmind, an AI company based in London. And they are perhaps best known for AlphaGo and a famous game against Lee Sedol. So Go is a very old Chinese game. It's a board game where you have two opponents using black stones and white stones. And the aim of the game is that you want to conquest as much territory as you can on the board. And what you see here on the right, this is Lee Sedol, a world champion in Go, playing against AlphaGo, the machine. The gentleman here on the left is an engineer from Deepmind, just putting stones following the algorithm, what the machine and AlphaGo is telling him. And this game is very famous because the thought in the community in computer science and mathematics is that we there was no hope to have computers to beat the best human players at Go for reasons that I will explain in a moment. So again, to understand the achievement on the Go side, let's step back and go back to the late 90s. This is a very famous game in chess between Garry Kasparov, a grandmaster in chess, and another machine, IBM Deep Blue. This is all, again, a very famous game because it was the very first time that the computer was able to beat a chess a grandmaster of the caliber of Garry Kasparov. Why it is hard to build computers that can play against humans, one way to understand it is the number of configurations. How many possible moves you can have when you play chess. And we think that in chess, we have roughly 10 to the 47. So like you put 1 and then 47 zeros. These are extremely large number. Despite this huge number, IBM engineers was able to write down the rules of chess and then have Deep Blue beat Garry Kasparov. This strategy of writing explicitly the rules of the game for a computer doesn't work for AlphaGo. And the reason is that in AlphaGo, if you ask yourself what are all the possible moves when you play a game, the number is 10 to the 170, extremely large number. And to give you a sense, this is larger than the number of atoms in the visible universe. So what you see here, imagine you have this configuration in the game, and you ask what are the possible outcomes starting from this configuration. You have this branching where each branch is a possible move. And there is a combinatorial explosion. So computer scientists tried to write down explicitly the rules of the game for Go to beat the best players, and they failed. And as I said, the reason is that we have so many combinations. So now Deepmind achieved this, again, tremendous achievement by using an alternative approach based on AI. So the idea now, instead of writing explicitly the rules of the game for the computer, you train the computer. You let the computer see many Go games, the game will be trained, and by training, the computer will itself learn the rules of Go. And it turns out that this use of AI and training machines is extremely important for proteins. So let me give you an analogy. The equivalent of IBM Deep Blue and the chess and the game against Garry Kasparov is this machine called Anton. See, this is a very sophisticated computer owned by D.E. Shaw, a billionaire in New York, who is working and running a company on protein structure prediction, protein folding, and drug discovery. So the idea of Anton, so this computer, is that you start from the rules of physics, because after all, proteins fold following the rules of physics, the way how atoms interact. So you start with your physical rules, you put them into the computer, and then the computer, using this expert knowledge, will predict the structure of your favorite proteins. And here we see the results coming from Anton, this sophisticated machine. In red are the experimental structures. In blue are the predicted structure by Anton. And as I hope you will realize, there is a good match. Anton is extremely successful at predicting protein structures. But what you may notice is that all those proteins are rather of small size. Anton now is incapable of predicting protein structure for proteins with a reasonable large size. So the analogy here is the following. Those small proteins, they explore a small combinatorial space like chess. Therefore, an expert algorithm with a powerful computer can fold those proteins. For larger proteins, they look more like the Go game rather than chess. So this strategy using an expert algorithm doesn't work. So we need to turn to AI and training to tackle the problem. And this is precisely what AlphaFold 2 did. They wrote down and they built an artificial intelligence system based on training. So I will try to give you an intuition about AlphaFold 2. This is a very technical topic. I will try to do my best to give you at least a glimpse and an intuition about this system. The starting point for the AlphaFold 2 AI system, and pretty much all the AI system, is the idea of coevolution. So you start with your favorite protein, which is a line here, a row, where you start with your sequence of amino acids, and then you search into those genomic databases for protein homologs, protein that are similar to your starting protein and that are related to your protein by evolutionary relations, and you generate what we call an MSA, and then you look at the variation along each column for amino acids. What type of variation evolution was exploring, and then you extract some correlation between those amino acids and their covariation. And the idea in a nutshell, if they are correlated, if two amino acids are correlated at two different columns, this suggests that those two amino acids are close to each other in space. And this allows us to fold proteins in 3D and predict the structure starting from the sequence. So this is the idea of coevolution beyond all the AI, most of the AI systems that we are currently using. So if you think about AI system in broadly and in the context of biology, in a nutshell, the idea is that you have an input. In our case, the input are the sequences of amino acids. You have a network where you turn the knobs to have a good training, and you have the output, the object that you want to predict. And in our case, these are the three-dimensional structure. So in a nutshell, AlphaFold 2 will start with a sequence of amino acids describing a protein, and then the protein homologs. So you end up with a list of sequences. You feed this list into a network, and then you output a three-dimensional structure. So this is in a nutshell AlphaFold 2. Now I will tell you a little bit more about how it works. If you don't understand the details, it doesn't matter for the rest of the my talk. So again, you start with your favorite sequence. So imagine that those tokens with different colors and shape are different amino acids. Imagine now that you search for homologs, proteins expressed in other species, bat, monkeys, so on and so forth. You generate what we call a multiple sequence alignment, and then you feed these objects, those matrices, into a network that will now try to extract correlations between different amino acids. And the color coding here means that amino acids are either highly correlated or they have a low correlation. So for instance, if I look at this entry in this matrix, it means that those two are highly correlated. And you let the network reason over and over and over again on those MSAs with the aim of extracting correlations between amino acids following the variations along evolution. So this is the first track of the network. The second network is really trying to encode the physics. Remember, as I told you earlier, unlike the chess, we don't write the rules that govern the protein folding and the protein structure. We let the network learn by itself those rules. So this track is about the physics. It's the pairwise correlation between atoms in the protein, and highly correlated atoms means that it's very likely that they come close to each other in space. And the network is making a reasoning over those relationships in space. So after reaching a sort of confidence about those correlations between amino acids, a network will use those representations to build a final three-dimensional structure. So this is AlphaFold 2 in a nutshell, and I'm happy to tell you more details during the discussion session. So now, where are we? And I told you at the beginning, the vision is first to close the gap between the amount of sequences available and the amount of structures available in publicly available databases. Now, using in silico prediction, an AI system, we are starting to build databases such as the AlphaFold protein structure database that is expanding by a large extent the amount of structural information. And the idea this year and Deepmind vision is to release hundreds of millions of structures in public databases. And again, this is a turning point for structural biology and biology. If you want to have a better understanding at high level of how AlphaFold 2 works, you can read our review with my colleagues Schrodinger and Al-Quraishi. On our front, we were extremely excited, as many people in our field, by AlphaFold 2. So we decided with a group of very talented students to reproduce AlphaFold 2 from scratch. So we have now our system equivalent to AlphaFold 2. We call it OpenFold. It's a fully trainable version of AlphaFold 2. So if you want to play with AlphaFold 2, the code that is currently publicly available can only allow you to make prediction. You cannot retrain the system and understand how the systems work. So for an effort to make the code available to the whole scientific community and beyond, we made our code available. It is on GitHub, and we have even the trainable version. And what you see here in blue is a protein predicted by the AlphaFold 2 system. And in red or pink, depending how you see it on your end, this is the very same protein predicted by our AI system, OpenFold. And as you see, there is a perfect match. So we are very excited that we were able to achieve this level of accuracy in our reproduction, and this is highly non-trivial. So a few words. What is our vision of OpenFold? Why we have reproduced AlphaFold 2, and what are we aiming to do with OpenFold? We are trying to tackle multiple conformations. Proteins don't have a rigid structure. They can explore different conformation. This is very important for their function. We would like to address docking, protein small molecule interaction. This is extremely important for drug discovery. Large multi-unit complexes. Often proteins, when they achieve their function in a cell, they don't act as an individual, but they come together and they form complexes with other proteins. We are also using our OpenFold for chemoinformatics applications and protein design. So this is, in a nutshell, our vision for the coming years with our OpenFold system. But now I should tell you a little bit about the limitations of the current AlphaFold 2. As I said, AlphaFold 2 is an extremely powerful AI system for making predictions and predicting 3D structures with high accuracy in most cases, but the system has deep limitations. So what I'm plotting here in the y-axis is the accuracy of the model. The higher, the better, 100 being a perfect prediction. And now what I'm plotting on the x-axis is the number of the depth of the MSA. How many evolutionary relationship you have found in your genetic search, because as I told you, the starting point for AlphaFold 2 is your target protein and its homologs. And what we see in this plot is that when we don't have much evolutionary information and protein homologs, there is a substantial drop in the model performance. So when evolutionary information is available, the performance of the model is really good. It's above 80 IDDT, which is really good. But below a certain threshold, we see that there is a substantial drop. And this is a major limitation for AlphaFold 2 with deep implication to biology, but also future pharmacological applications. So let me tell you now our alternative approach for doing protein structure prediction using a single sequence and tackling the limitation of the AlphaFold 2 AI system. So this is our system. We call it RGN2 for recurrent geometric network. Our paper is available if you want to read more about our system. If you want to have a high perspective about our system, you can read this nice piece in Science, in the journal Science, by Derek Lowe, contrasting our strategy vis-a-vis the strategy taken by AlphaFold 2 Deepmind. So why we want to have an alternative strategy for tackling the limitation that I mentioned? So first of all, we would like to predict orphan proteins, proteins for which we don't have evolutionary information. We would like to predict the structure of rapidly evolving proteins, such as the virus that we are suffering from and the spike protein. We would like also to explore design structures. We have the ability to design proteins that have not been seen by evolution, and we would like to have a rapid exploration for such proteins. And we would like also to understand the principles that govern polypeptide folding. And the current AlphaFold 2 AI system is not able to answer the questions that I have listed here. And we believe that our system, our alternative way of doing protein structure prediction, could at some point answer those questions. So in a nutshell, in the remaining minutes, I will briefly guide you through our system. So our system is based on two novelties. We have a protein language model and the geometrical module. And the protein language model, again, is inspired from tremendous development in AI coming from natural language processing. So now, for instance, if you use your mobile phone, you can easily translate between different languages. This is mostly based on AI systems. And now we have remarkable systems as well that can write prose and text as good, if not better, than humans in many cases, such as the GPT-3 system by OpenAI, and we have other systems in the natural language processing world. So inspired by those approaches, we decided to reason on protein space and sequence space using a similar strategy, and we call it a protein language model. And the idea in the nutshell is the following, is that now instead of using evolutionary information, as AlphaFold 2, we start with a sequence of amino acids, and now we use a common strategy in natural language processing, which is masking. So imagine you have words or amino acids, and you mask certain amino acids or certain words if you are dealing with text, and then you ask your network, in this case, we are using a transformer, this is like a cutting edge architecture in machine learning, to recover the missing amino acids or the missing words. And by doing so, by forcing the network to recover what have been masked, the network will build very powerful representations that account for the correlation between amino acids or words if we are dealing with text. So our system that we call AminoBird generate extremely rich abstract representations that you see here. I mean, this is a visual that captures both the nature of the amino acids, but also captures their relationships with their neighbor and amino acids that are far away. So this is our starting point. So now our AI system, again, is not looking at evolution. It starts with those very rich mathematical representations. And now let me just briefly give you some results. So when we look at proteins for which we don't have homologs, the proteins for which AlphaFold 2 system was suffering, I told you there is this limit where AlphaFold 2 performance drops substantially. So if I look at those proteins, one example here I'm showing, what you see here in gray, this is the experimental structure, this structure observed by experimentalists. In yellow, this is the AlphaFold 2 structure, and in purple, this is our own prediction, our own AI system. And as you see, we do better than AlphaFold 2, and AlphaFold 2 is even hallucinating regions that do not exist. So this is another case for humanly designed proteins. So the protein that you see here was not explored or discovered by evolution. It was designed by human experts, and the structure is in gray. Our system prediction is in purple, and AlphaFold 2 is in yellow, and we do better. So to sum up, there are situations where we don't have evolutionary information and a humanly designed protein where we do better than AlphaFold 2 in many cases. And we are very excited about it. As I said, we believe that our system is a complementary AI pipeline for doing protein structure prediction. One important thing about our system is that our prediction time is very fast. We can predict structures in milliseconds, typically, whereas AlphaFold 2 systems and similar, they predict structures in seconds. This is very important when we want to explore proteins at large scale, and we want to sample proteins at large scale, having fast pipeline. So in the last few minutes, I will conclude with a sort of vision. Now that we have those very powerful AI systems that can allow us to predict structures with high accuracy in most cases, what is the vision for the future, or one of the vision. I will now go back to the past and to the pioneer of our field, biophysics and mathematical biology. And I will turn to Erwin Schrodinger. I believe many of you are familiar with Schrodinger. He's one of the founding father of quantum mechanics. We know the Schrodinger equation. So Schrodinger wrote this beautiful book, What is Life? Based on a lecture he gave at Trinity College Dublin in the 40s. And his answer was about genomic information. So for him, the most important question about life is how is the information and the genetic code stored? And Schrodinger came to this amazing conclusion that the genetic code needs to be stored in an aperiodic crystal that we call today DNA. And his vision really inspired the founding father of molecular biology, including Francis Crick. But from our modern understanding, we know that Schrodinger's view was rather narrow. It is true that understanding genomic and genomic information is extremely important for life, but that's only one aspect. At the very same time, von Neumann, polymath and somehow the father of modern computer, was thinking mathematically about self-replicating machines. And starting from very abstract principle, von Neumann realizes that to have machines that self-replicate, they need to have two modules. An information storing module, a tape, this line, you can think of it as like your DNA or genomic code, or you can also call it a software, but you also need an information processing module, a machine that can read the code, the tape, and then replicate both the code and the machine itself. And this information processing module needs to be independent from the tape that stores the genetic code. And those machines today, you can call them proteins because they read the code and they do pretty much all the function in an organism. And also, you can call them hardware. So from our perspective, von Neumann distinction is between software and hardware. So in the last 20 years, there was a major development in biology after the Human Genome Project, and the development was mostly concerned by the information storing module, the tape. Now I believe we're entering an era where we could, at large scale, start understanding and exploring mathematically, computationally, the information processing module using AI systems. And the combination of large-scale computational, quantitative, mathematical models that can allow us to tackle and understand the genomic code, the information storing module, and the proteins, the information processing module, can lead to this vision, which is having a virtual cell. A cell is made of proteins, nucleic acids, and their interaction. If we are starting now to build AI and mathematical systems that could describe those components and their interaction, we could imagine that at some point we can simulate a whole cell in a computer. And this is an amazing vision, both for our understanding of life at the molecular level, but also for therapeutics, pharmacological discovery, and medicine, where we could imagine having experiments in silico in a computer. With that, I would like to thank you for your attention. And I would like also to thank the people involved in our AI system, the talented master students that worked on OpenFold and our funding agency, DARPA. Thank you very much. Yeah. 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 First of all, I think there are many ways, right? So one thing which is often the first step, say in drug discovery. So currently, for instance, one first step in pharmacology is screening. So imagine you have a target protein. You have very strong reason to believe that a given protein is involved in a certain disease, and you would like to inhibit this protein. So you will start by designing a chemical compound that will target this protein. So the way how it is currently done in most pharma company, you will run screenings at large scale. So you will take many candidates chemical compound and screen them against your protein of interest with the hope that one of the compounds or some of the compounds will target your protein. Now, if we are starting to have very accurate in silico system, instead of having those screenings, again, they are expensive, tedious, they take time, you can do these kind of screening in silico in a computer, because the computer can take many different compounds and test them against your protein of interest. And that's why I mentioned in our AI system, the time for prediction. Having fast pipeline is very important to get those, for instance, screening at large scale. So that's, I think, only one aspect of the possible application, near future application of those techniques. But I think it goes beyond that, because again, now often diseases are not related to an individual protein. They are involved into pathways, and we don't yet have a full mechanistic understanding of those pathways. So we know that certain proteins are involved in a given pathway, but how they form a complex, under which condition, we don't have a good knowledge at this stage. So having a structural knowledge, a knowledge based on those structural underpinning could help us to understand better how those pathways work and how some pathways could lead to dysfunction in a cell. So I think there are many, many opportunities where those AI systems could be applied for pharmacology and disease treatment.
Yeah, thanks. And one question, because you talk about the evolution. Recently there are a paper, probably in Nature, like last month, 2 months ago, that show that the mutation in the DNA are not random. It means that there are some proportion of the DNA that will be highly subject to the mutation and other not. And you show and you discuss this about the proteins, like to provide the information, the structure of the protein.
Yeah.
Do you believe that the machine learning, deep learning, or artificial intelligence will maybe in the close future provide us more information about the evolution, like if it's possible to decide between Lamarck and Darwin or not yet? Or is it something that will be still in discussion?
Yeah, I think regarding this question, and this is a subtle question, and the way how I see it is the following. The current AI system, they are very powerful for making predictions. Like the system that I explained is really capable of predicting structures in many cases with good accuracy. But those systems now, they act as a black box. We don't really know and understand what sort of computation they are doing and what they are learning exactly. So broadly, I don't think they are at the stage of answering questions about principles, because we don't understand how they work. They are very powerful for making predictions. And that's why all my examples are about predictions, predicting protein structures, predicting a docking pose. But as for answering those type of questions, guided evolution, Lamarck versus Darwin, I don't think they are, at least currently, capable of addressing those questions. And again, the reason is we don't understand how they work. They are a bit like a black box, and that's not only about biology. This is the case for all AI systems. So for instance, if I go to the Go game, which is in a way easier than protein folding, because we humans, we play, or at least some of us play Go, we don't really know what the kind of network have learned. What we see the outcome. We see that AlphaGo, for instance, is very powerful. It can beat all the humans. It's extremely powerful. But how it learned those representations and what is the meaning of those representations, that's something that we currently don't know.
Thank you,
But it's a very active research area in machine learning.
Yeah, I think there are several maybe question and discussion, but many thanks for today. And I will
Thank you very much.
our PhD were very interested on this topic, and they wanna be in touch with you after the seminar that you gave at the M6P seminar series. So thank you very much, and appreciate your time.
Yeah, sorry, I have to go. Sorry for the other speakers. I can't stay. It's very early, but thank you again for the invitation. I wish you a good day and a successful day in Dubai. Bye-bye.
Thank you, appreciate it. Thank you very much. Tarek Dauda, you are the second speaker today about the artificial intelligence to accelerate research in immunology. And many thanks, Tarek. You're welcome at the M6P and at Dubai Expo to present this nice topic. And please, if you can make it in 15 to 20 minutes, if it's possible. Okay. And after I will give the floor to Sibrahim. Thank you very much, Tarek. You're welcome. And if I will present you just in a few. Tarek Dauda specialized in application at the intersection of artificial intelligence and immunology. He did his postdoc at Harvard Medical School and the Broad Institute. Broad Institute, it's an institute between MIT and Harvard. And he joined the M6P recently to develop this kind of topic, the application of machine learning for biology questions and medicine also. Tarek Dauda, you're welcome, and thank you very much for your time today.
Thank you, Rashid. I hope everybody hears me fine. So, thank you, everybody. Welcome, welcome to. I wish you a very good evening, everyone. So, I'll start with the general history of what we do as human beings. So we as human beings, we make tools. Historically made tools to increase our power, we make tools to increase our speed, our perception, and we also made tools to increase the capabilities of our mind, to increase our memory, to increase our computation abilities. But what's missing now is our interpretation abilities. We don't really have tools at the moment that help us interpret things. And interpretation is something that's really hard because it's loosely defined. So we have two phenomena, phenomenon A and phenomenon B, and really what we're interested in is how are they linked? What's the story behind these two phenomena? We like to derive meaningful and actionable knowledge from the relationship between A and B. And this has become like a very big problem now that we have big data. And the goal of this presentation is to introduce how we can use AI to basically augment the researcher, to enable the researchers to make more discoveries faster. So this is the title of my presentation, How to Accelerate Research with Artificial Intelligence. And I would like to thank all these different institutions that participated to the works that I'm going to present. So our first example here, where I'll show you three examples. So the first one is about how we can understand the origin of cell markers from mRNA sequences. And for these markers, I mean MHC-associated peptides. So these are MAPs for short. So these are small bits of proteins that are basically made inside the cell and that the cells present at the surface. And these are very important. They're like cornerstones of the immune system because, for example, if the cell gets infected by a virus, then the cell will produce viral proteins and then will show at the surface some peptides from these viral proteins, and then the immune system checks the cells, and if they find a cell with a peptide from an abnormal origin, they just kill the cell. So this is how the immune system is able to detect cells infected by parasites, viruses, but also cells that have become cancerous. Of MHC-associated peptides is the following. So you have mRNA sequences. These mRNA sequences are translated to proteins, and then these proteins are cut into small bits that we call peptides, and then some of these peptides will be presented at the surface with the MHC-1 molecules. So prior to this study, the predictors that we have had a high false positive rate. So that means that we will flag a lot of these peptides as being presented where they were not presented. So that tells us that there must be another explanation, and we went on looking for that explanation. And our hypothesis for that was that it's the translation, the mRNA translation when we go from mRNA to protein that we haven't looked at that can explain this this phenomenon. And it makes sense because translation is a very, very complex process. So you have this very big protein machine that's called the ribosome that goes and reads the sequence of mRNA and translates it into a protein. And to do that, it uses tRNAs. So we have more than 600 tRNAs. The mRNA is read basically three letters at a time. So we call them codons. We have 64 codons, and in the end only have 21 amino acids. So what that means is that there's actually a lot of information, a lot of things happening before we get to the 21 amino acids. And the question was, is there something here in these 64 codons that can explain? So we went on to answer this question. And so first of all, we needed data. So we extracted 120,000 sequences associated to these MHC-1 peptides. And then we compared these two types of sequences, the original mRNA sequences and the translated protein sequences. And we gave them to two neural networks that are basically identical neural networks. And we asked them to predict whether these sequences, because we have in these sequences, we have sequences of map origin and sequences that are not of map origin. And we asked the networks to basically make the difference between them. And this experiment can only go into two different ways. So the first one is that the mRNA sequences are better. So the accuracy is higher when we use mRNA sequences, the original one, not the translated one. And the second one is basically they're the same. And the reason why this is the case is because mRNA is translated into proteins, and we have 64 codons, 21 protein amino acids. So it means that if the accuracy is higher with mRNA, it means that basically the codons encode information that the amino acids do not. And so these are the results we got. So you can see like the original sequence here is in blue. The amino acid sequence is in green. And here on the x-axis you have the size of the sequence we gave to the networks, and here you have the accuracy. And you can see that for all different sizes when we give the mRNA sequence, and that was the first big result of this of this study, that mRNA actually has an influence on the presentation of the MHC-1 associated peptides. And this is something that we didn't know before, because all the research, the predictive research before that was entirely based on the protein sequences. So then we went further, and this is when it gets really interesting. So we opened up the neural network and asked it, what are basically the features of the sequence that make you decide whether it's going to present a map or not. So, and this is what we can see here. So on the x-axis, you see the sequence. So and for every one of this position, you have what we call the preference of the network for a given codon. So codons again are these three-letter words that are then translated into amino acids to make proteins. And you can see that even though, for example, here, this is serine. So serine is an amino acid that can be translated by 6 different codons. And even though these codons are synonymous, they translate to exactly the same amino acid, we see that there's slight differences between them. Same thing for prolines here. And we can see that proline and serine are basically opposites. And when you have a serine close to the region that will become a map, which is that having a serine, like a codon for a serine close to that region, increases the preference, which means the probability that the network will classify this sequence as producing a map. And we have the opposite here. So this decreases the probability if you have a codon that encodes proline. But things get more interesting when you look at other amino acids. So for example, here we have tyrosine. So tyrosine can be encoded by 2 different codons. So we have TAC and TAT. And what we see here is that although these codons are synonymous, which means they give rise to exactly the same amino acid, they have opposite opposite effects on the presentation of the map sequence here according to the network. So if you have a TAT, the probability increases. If you have a TAT before the sequence, the probability increases. But if you have a TAC, before the sequence, then the probability decreases. Although they encode exactly the same amino acid. Leucine here is even more interesting because we have more codons, and we can see that 2 codons here for leucine are basically neutral, so they don't change the presentation probability. However, CTA increases it, while the other one, CTT, TTA, and TTG decrease it. And so these are very, very interesting results because for the first time, we have an hypothesis on what codons where can affect the presentation. So then we decided we had that, and so the next experiment that we ran is basically we wanted to see if now that we have these results, so we see that the mRNA sequence seems to have a very strong influence in modulating the presentation of MHC peptides. We want to see if our predictor, so we built a predictor called CAMAP that uses this information, can increase the accuracy of what we currently have. And so we compared CAMAP with with the best previous metrics that we had. And the first one is what we call the ligand score. Ligand score is basically how likely the peptide is to stick to the MHC1 molecule, and then the expression of the mRNA. And we can see that when we add CAMAP here, we always increase the accuracy very, very significantly. And we tested it on different data sets. So this is the data set, the original data set, human BLCLs that the network was trained on. And you can see that the accuracy is higher on this one. But what we did here is that we took exactly the same network. We didn't retrain it. We took the network training on BLCL and then applied it to different data sets. And this is not something that's usually done. Usually like a network is trained on first, and then it is used on that task. But for this experiment, we did something unusual. We trained on one cell type and then applied it to all the other cell types that we had. And we can see that even though the network was trained on only this cell type, it actually can make accurate predictions on different cell types in human and also on cell types in mouse. So what that tells us is these results are actually very, very strong results. And finally, for MHC1 peptides are very important when it comes to the immune system. They're really this cornerstone of the immune system. And so we want to see if we can help ask like this very important question of the false positive rate. Can we decrease the false positive rate with our new predictor? And the answer is yes. And so we went from basically 33% false positive rates, which means like peptides that are flagged as being presented but never presented the surface really, to just 1%. And this is for the top 1% hits, which are the peptides that you're more interested in if you're going, for example, to make a vaccine against a virus or a vaccine against a cancer. These are the ones that are really of interest to you. And we were able to dramatically decrease the false positive rate here using our predictor. Validation. This is some very interesting and very exciting work. So we use the network, the figures that I showed you before, where we see like the different preferences, and using that network, we designed sequences that according to the network, we present a peptide a lot, or like higher, we generate a higher presentation of that peptide or less presentation for the same peptide. And you can see here, so the blue one is the one that was predicted by the network to generate more of that peptide. And you can see that it's actually here, and the prediction of the network is actually true. And the same thing for the red one, and the red one is the one that was designed to generate less. So this is very exciting because we were able, using AI, not only validate the results of our predictor, our neural network predictor, but we were able to design genomic sequences informed by the network that we've trained earlier. So in conclusion, for that part, we were able to link mRNA sequences to map presentation using 120 sequences of mRNA sequences, and we were able to demonstrate that mRNA modulate map prediction presentation, and we're able to design sequences using AI, and we also were able to reduce the false positive rate for the top therapeutic targets from 32% to 1%. So this study is both hard fundamental research and applied research at the same time, because the implications for this is for vaccine design against cancer, viruses, and parasites, and also it also has an implication about understanding, giving us a better understanding of autoimmune diseases, because these peptides might be linked to some of these diseases that we don't understand at the moment. So the second presentation is about how we can get higher definition human cells from different types of sequencing. So up until very recently, we had, when we talk about sequencing, we talk about bulk sequencing. And bulk sequencing is you basically take a tissue and you just like a piece of a tissue and you sequence everything at the same time. So this is basically the difference with what we have now. Now we have a single cell sequencing, so we're able to sequence the cells one by one. So this is basically the difference between a smoothie and the cell salad. And the fact that we're able to sequence the cells one by one means that instead of having like an average of the tissue, we can really get every one of the cells and really get into the details of what that cell is doing. And so this is very, very exciting at the moment. This is one of the frontiers of biology, and a lot of research is being done. It's being done on that topic, and we have different kinds of sequencing of single cell sequencing. We can read the DNA. We can read what we call post-translational modifications, that things are added after the mRNA has been translated to protein. We can sequence the mRNA, we can sequence the protein, we can sequence markers of the surface of the cell. And the idea is basically to take all these different types of sequencing and integrate them into a single map of the cell that's very accurate and tells us exactly what the cell is doing at a given moment. And this is an example with mRNA and protein. So mRNA and protein are two different things. So mRNA is basically translated into basically the blueprint for proteins, because the proteins are translated into mRNA. So mRNA is basically what the cell is planning on doing, and the proteins are basically what the cell is doing at the moment. So the proteins are the small machines that work inside the cell. And so if we take mRNA and proteins and integrate them together, we get this map. Like if we just look at the mRNA, there are some very important cell types here defined by immunologists like CD4 and CD8 T cells that are very, very different when it comes to the immunological response. But when you look at the mRNA alone, we're not able to differentiate them. However, if you look at the protein, you can see that they're very, very different here. And same thing goes for different cell types. And by integrating both mRNA and proteins, we're able to create this map where everything is integrated together. And you can see that CD4 and CD8 are very different, placed differently, and as expected, basically. And so this is the demonstration of what a high definition integration looks like. So you can see that, so this is, these are millions of cells that have been integrated together with different types of sequencing. And you can see that every color here represents a cell type. And after the integration, you can see that the different cell types are really clustered together. And even the transitions, for example, here you have the B cells. B cells go from naive to intermediate to memory B cells. And you can see that the tree, like the red to the blue to the purple here, you can see like the transition of the cells. And so this is like an incredible tool for biology at what the cells are doing at a given moment in time. So in conclusion for that part, so we were able to, by using artificial neural networks, we're able to take different kind of sequencing, mRNA proteins, and mRNA proteins, etc., like, and aggregate them, integrate them into a high definition image of the cell. So that gives us like an accurate cell representation. It allows us to identify new cell types, and this work literally has implications for everything from cancer research to autoimmunity to research in biology in general, because if we are able to get a better vision of what cells are doing, this can have applications and implications for basically anything that's linked to biology or health research. Finally, we have this work on viral transition markers. So we know that viruses can move from one species to another. Here we mainly interested in influenza. So influenza can move from different hosts, from avian to human to swine, back and forth. And what we're interested in is to find the markers that we will tell you basically this strain is likely to move from one. And usually. So what we did is we trained a neural network with using 200,000 cells, mRNA sequences from influenza. And from these sequences, we trained the network to basically predict the host. Is it human, avian, or swine? And we got a very high accuracy. But for this study, we did something a little bit different. So instead of just being content with the accuracy, usually this is what we do in machine learning. You have a very high accuracy, you're done, you did a good job. Is basically to look at the mistakes that the neural network has made. So if you have a 90% accuracy, the question was, what can we learn from the remaining 2%? Because if your network is very, very accurate at predicting something, here the host of the virus, basically, if the network is predicting for a given mRNA sequence a different host, what it's actually telling us is probably this sequence is more like, it's not a human strain, it's more like an avian strain. This sequence has features that I would normally associate with an avian strain. So these are a map of the results we have. So we trained two different kind of neural networks, state-of-the-art neural networks, so bidirectional STMs and transformers. And so you can see like, here is a strain, and you can see that they're very well clustered. You have for RNA and proteins, you can see the frontier between the different different species, and the high here you can see that the more blue it is, and bigger the mistake the network made, which means that they gave a higher probability to a different class. And these are the sequences we are interested in. The sequences here are the frontier between the different species. So we extracted these misclassified sequences and we analyzed them individually using a neural network interpretation tool called LIME, statistics, and the information theory. And by taking all these methods together, we're able to extract the markers that we're interested in. And so this is what it looks like. So with our results, these are the results we have. These are the markers that we have. And we contrasted them with what other people had found before using biochemistry and not in silico method like we did here, but like real biological experiments. And so you can see here, this is what we found, the intersection to what we found and what the other studies found, AG, they call it AG and they call it CTG here. And same thing here for, and same thing here for another study. And when we put them all together, we end up with this AGA codon that's actually a very important codon for human transition. And we went even further. This is something that wasn't done before. We looked at the position for each one of these markers. And here you can see the different positions that we found for the transition from human to swine for the human PA protein of influenza. And these are very, very interesting results and very important because they can, it's not just AGA somewhere in the sequence. Now we can really tell, okay, this part of the sequence, if you have an AGA at this region, then it means that it's probably this strain is likely to transition from a swine from a human to swine. So in conclusion for this part, we went from influenza mRNA and sequences to transition markers, and that allows us to identify strains that are likely to transition between different species, and this has implication for public health and agriculture. So to wrap up here with the story of humankind, AI is basically for that will allow us to increase our ability to interpret, to link phenomena together, to speed up research. And I gave 3 examples in this presentation, how we linked mRNA sequences to cell markers, how we link different types of measurements to get a higher definition of immune cells, and how we analyzed influenza mRNA sequences to get the transition markers. And that basically gives us unique opportunities, especially when you're talking about the African continent, which is a very diverse continent, but also a very understudied continent. And by using these techniques with AI boosted research, where AI is not used like a simple predictor, but really like a tool that is integrated fully and completely into the research process, we can move faster and solve important issues faster for public health, agriculture, or any other subject that we think are very important. So that's the end of the presentation, and I would like to thank all the people who participated to all the work that are presented, and all the institutions that participated as well. So thank you very much.
Tariq, thank you very much for this nice talk. Thank you very much for your time, and thanks for this free, important scientific project that you presented. And I would like just to ask you 1 or 2 questions. It's about like the high definition that you presented, like let's see, take the example of peptide sequencing in the case of COVID. So is it possible in the future, based on the machine learning, like to predict next mutation that can happen, or it's still not possible?
I think it is possible. Yes, I think it is possible. In this work that I presented here is basically the precursor for that type of work. Here we worked on different species because it's easier, because it's more defined than having like different strain of the same virus for the same host. But this is the first kind of work, the easiest basically to start with, and then by applying the same kind of techniques and probably refining them a little bit, we might be able to predict how a virus might evolve in the future.
Yeah, and about the immunology part, and as you have like this single cell sequencing, is it like a beginning of personalized medicine? Like, is it something that can be done in the next future?
Yes, yes, yes. I believe so. I believe so. This is like not at the moment, but in the near future probably, because as the cost of sequencing decreases, we will be able very soon to have single cell sequencing of individuals in clinical setting. And if we have these models that are already trained at integrating these different modalities of sequencing, we can sequence different parts of the same cells and then integrate them into something like this that hopefully will look better, because it will be like in the
Yeah.
future. This is still research that clinicians could look at and derive hypotheses about what's happening, basically what's the disease doing at the moment. And this has very important implications, for example, for cancer, because we know that the environment, the direct environment, immune environment of a cancer is very important when it comes to remission and recovery. So we would be able, clinicians would be able to look at these immune cells that are surrounding the tumor and base their diagnosis and prognosis on basically these immune cells, what they're doing at the moment, and what type of cells are present or not present around the tumor.
Yeah, thanks. And maybe my last question, it's about like the perspectives in the research that you developed before and now for the African continent. And you showed a beautiful picture, unique challenges. Which one you would like, you wanna like take and to do it like for some impact on African population and of course Moroccan population?
Yes, so there are many different challenges. There are challenges when it comes to metabolic diseases that are understudied in the African continent. There are also challenges when it comes to parasites and infections, because Africa is an extremely diverse continent. We have deserts, we have equatorial forests. There's a lot of things happening in Africa, and there's a lot of parasites. There's a lot of diseases that we don't know of. There's a lot of also genetic diversity that we don't know of, because the African population has not been as much sequenced as, for example, the European continent. So when it comes to the diversity of, for example, the MHC1 molecules that are very important for presentation of markers of pathogens, we know very little for some regions of Africa. So these are all different domains that we could study using AI. And also there's also I just talked about the public health aspect, but there's also like all the agricultural aspect, like when it comes to livestock and agriculture in general. There's also a lot of things that we can do to study, like the different parasites, the different strains, to basically increase. We could do a lot of things to increase the output, for example, of agricultural land using AI boosted research.
Tariq, thank you very much. I appreciate it. It was nice talk with you today, and it was great. Thank you very much. And we'll completely maybe now change the area. See Ibrahim Laqsir. He will describe the embedded artificial intelligence device for medical application and the case of rapid COVID-19 ER portable sensor for mass screening COVID-19. See, Ibrahim, thank you. And I think the floor is yours. Ibrahim Laqsir is the director of the Digitalization and Microelectronics Smart Device Center at Masir. UM6P, as well experienced with 26 years in microelectronics, semiconductor industry research and development design, assembly and test new product package development, etc. Ibrahim, you're welcome today, and thanks for your time. Unfortunately, you are not with us today at Dubai, but it's from Casablanca, probably you are now.
Thank you, Rashid. We hope to be with you. So did you hear me?
Yeah, absolutely. Next time, we will do it next time. I think
Okay.
you'll do it next time, inshallah. Thank you very much.
So I share my screen.
Yes, please.
Is it okay for you?
Yeah. And we will end today by Professor Gabriel Malka after your talk. Thank you, Ibrahim.
Thank you, Rashid. Hello, everybody. I start my presentation. First, I will present rapidly the MASIR. MASIR is the Moroccan Foundation for Advanced Science, Innovation and Research, created in 2007, and was created with 3 poles. One is microelectronics, biotechnology, and materials and nanomaterials. And we have more than 700 scientific publications, around 200 patents and different prize, and we are running, I think, more than so our the strategic orientation for MASIR is to support the national strategy and plans, and also to support Moroccan company to move up into the value chain, technology transfer, and promote growth locally through complementarity and synergic. So we have, as you can see, different partnership and contract with the different public and private company. The core R&D of MASIR is in the biotechnology. We have the medical biotechnology and the green biotechnology. For the material and nanomaterial, we have a composite and nanocomposite, engineering and durability of material, development of the natural resources, optics and photonics. For the microelectronics, we have the 2 departments. One is the embedded system and the IE, and digitalization and microelectronics smart device. So as we are in this medicine of the future, I need to share with you at least some typical projects that have been ended or developed at MASIR regarding health. We have already developed what we call a multi-parameter monitor with the smartphone. As you know, MASIR have been involved also in developing the RT-qPCR test. And up to now, we already sell more than 3 million kits for the Moroccan Ministry of Health. We have also another point of care, what we call the mobile care for tuberculosis, diagnostic and screening. So this device have been developed between the 2 departments in biotechnology and microelectronics. And today we are in the phase of going to the market and also the CE marking review. We have also some kits that have been developed for the breast cancer, hepatitis, and leukemia. And we are on another project for the microfluidic kit. So the ongoing development project is the COVID influenza diagnostic kit, the COVID UK variant diagnostic kit, rapid antigenic and serological test, prostate cancer kit. Okay, and we have also obtaining some registration certificate for reagent for in vitro diagnostic use, like the SARS-CoV-2, hepatitis C kit, breast cancer, the SARS-CoV-2 with the triplex, tuberculosis kit, and leukemia kit. The ongoing device that we have is the microfluidic chip, the lab-borne card, multi-parameter monitor with smartphone, and rapid COVID ER. So the finalized device is the lab-borne card and the rapid COVID IR. And also we have in the pipe some project on the telemedicine and also some listening of the Ministry of Health project that we are on the way to develop. So for the center, the digitalization and microelectronic smart device center, Smart Device Center of the Moroccan Foundation for Advanced Science, Innovation and Research is specialized in the design, simulation, and packaging of microelectronic device, assembly and test of surface mount technology, as well as material characterization. The center's facilities is equipped with a semi and fully automated assembly line capable of producing few prototype to medium volume of microelectronics device. The platform also offers service in demand of failure analysis and reliability test for national and international partners. The center is involved in applied research in different programs requested by industrial partner, fabless company, and SMEs in the area of sensor, chip packaging design, and system hardware and software development. So the expertise of our team is mainly in package and PCB design, software development, system integration, IoT, chemometric research, machine and deep learning, big data analysis, and artificial intelligence. So regarding our talk today is for the what we call the rapid COVID IR overview. The rapid COVID IR electronic is an electronic device that allow a screening of absence of SARS-CoV-2 with a nasopharyngeal sample with a sensitivity rate of 94% and a specificity rate of 70%. The testing result in less than 20 seconds. The device indicated in the logic scale, large scale screening has many advantages like no specific preparation required, no reagent required, fast, and also a very, very small device. So this device uses a technology based on near-infrared Fourier transformer spectroscopy, which allows spectral data to be collected simultaneously over a wide spectrum using the miniaturized Michelson interferometer principle. So it also has built in hardware to embedded artificial intelligence model after processing the result specific spectral image. So the connection between the sensor and the mobile application is ensured through a Bluetooth communication mode. Once the server receives the data, algorithm processing is performed using a various predefined model. Spectra are stored on the server for further analysis and improvement of the artificial intelligence prediction model. So the device is controlled by a mobile application pre-installed in the phone, supplied and also downloaded from Google Play Store. So as during the pandemic COVID, we start developing with three phase this device. So as you know, we didn't have access to the sample during the COVID. So what we do is we work with some labs, medical labs, to perform three things. First, the data acquisition, the development of mathematical prediction model, and the clinical validation. So the first step was used to build a database of sample, something like 1,500 positive and negative COVID by a nasopharyngeal sample tested by the reference method, which is PCR, and scanned by the rapid COVID IR device at a medical analysis laboratory. So the development, the second phase is the development of artificial intelligence mathematical model applied to the previous database to identify specific signature of the absence, presence of COVID test with an embedded AI on the device. And the third phase was the validation of the device carried out of the reference laboratory on 585 COVID sample according to the standard NF 15189. So here you can see the architecture that we have developed with this this device. This is a remote sensing device also, and here you can see also the confusion matrix and statistic reference prediction value that we have found in terms of sensitivity, accuracy, and specificity. So the prediction using machine learning model in the first many prediction machine learning model were tested to predict negative and positive case. The interpretation of these model allow to find the pattern which separate the case and also helps to select the band and wavelength which are linked to the variation of the presence of coronavirus. So as you can see here, the tool for variation selection has been used to select the detection, the band and the wavelength are linked to the coronavirus. So we didn't care much to the spectra, but to those fingerprints that can do the selection, the variation between the positive and the negative case. And as you can see here, so if you can see here, the positive sample and the negative sample, we have a clear segregation between the negative and the positive one. So after we use what we call a deep learning, and you can see here the architecture of use deep learning model with the same technique, but we have more data and more, I think, more than 2,000 sampling on which we apply this for the input layer and the output layer, positive and negative case. So the theoretical result to this deep learning model, you can see here, we reach on more than 1,700 sample. We have an accuracy of 99%, and for the test data set, we reach on 430 sample. So I think here we have 144 positive and 286 negative, and the result was around 92% of accuracy. So the device, as you can see here, is this one. And today we already start so the spectrum is this one, and we start using and selling this in Morocco since last year. And today, I think we have a very, very accurate for mass screening for the COVID. So we start using the same process of taking the nasopharyngeal swab according to the usual procedure, and plus we use the same procedure as the PCR. So here you see the certification for the Moroccan lab. because to use this in Morocco as a medical device to screen the COVID-19. We can also show you just some number regarding a period of, I think, 2 weeks or 1 week using this device as an industrial partner that use this mass screening. So we screen around 5,000 person. They found 600 of them suspect, and when they take this person to the PCR, they found 61% positive by PCR. And the deployment today in Morocco, we have tested more than 200,000 tests performed in Morocco. We also fine-tune our model for the Omicron variant. So I need to thank the team who was involved in this project. So today I think we have eight engineers and researchers working on this project. The most important thing is when we face to this kind of virus, we have to try to find a very fast solution for testing mainly. And thank you, Rashid, and thanks to all the team.
Brahim Al-Qasir, thank you very much for your time and this interesting presentation with this very important device. And I would like to thank you, and I would like to ask you 1 or 2 questions. It's about the specificity and the sensitivity of this device compared to the RT-PCR. Which one is more sensitive?
The device have been developed to be a reference for PCR, and the sensibility is 94%. So for the portable medical device is something that can be. This is why we receive also the certification, because 94% is for PCR, and the specificity was low, was 73%. But the difficulty we face is mainly to different variant. For example, when we have developed this is for the Delta variant, and each time we have to, when a new variant appeared, we have to use a different model. Okay.
Okay, see. And one question, like you did 200,000 tests, right? So are you still improving your data, your algorithm, your system by using all of this data that you are accumulating, or it's
Yes.
you would like to develop a new device in the future or something that based on all the data that you have now?
So yes, I agree with you, because it's today it's a remote sensing device, even if it is on the hand of on other partner, but we have the data, and each time we have some data, we improve by more learning model, and today we are in the version of the sixth model that we have developed, and we improve the data. For example, for the sensitivity and specificity I talk about, but after improving more data, we reach more 98% of sensitivity and more than 85% of specificity.
Okay, and yeah, I don't know if there are any maybe data on that, but I am curious about, like you compare sample with COVID particles and sample without COVID particles. Both give you like a spectre, and the spectre that you get from the samples with COVID seem to be different, right? From the samples without COVID.
Sure.
And why? Is it like RNA or some specific protein, membranes, receptors, something in the COVID particles, variant, that give this change in the spectra that you have, or you don't know yet?
So we practically we show that we have some difference because some protein, I think, like Tariq or Hamza present, some protein react differently. I think the chance we have is we don't test the RNA, but we test the virus as it is.
Okay.
This is the most important thing.
Okay. Okay. Interesting anyway, because it's strange. It's strange for me. Like they have you get some spectrum at very low. I think, I don't know, in the samples. I don't know how much particles are there, but it's promising, and I hope this device will help us because the COVID is still going up. It looks in China is going again up, even if we don't want to hear about the COVID anymore. But it's still there, and it's still going, and I hope it will not get some bad mutation. And congratulations for this device. It's very important for our country and for the African continent if we can provide some nice and important research and development for the country. And this one is one case, and I hope in the future you will be able to propose additional devices for COVID and other diseases. Ibrahim, thank you very much.
The idea we have, Sir Rashid, when with the COVID appear, we have some state-of-the-art paper, maybe in Brazil, that the spectroscopy coupled with the IA can detect in the labs what we call the Zika virus.
Okay.
And today, the opportunity we have is to work with the same device and with the same expertise to develop, to address other virus. This is what we are doing now.
Yeah.
Thank you very much.
Yeah, it's very interesting. Maybe if you compare different viruses and maybe a device to give us idea about the contamination, or what kind of contamination maybe it can be with a huge applications. Ibrahim, thank you. Thank you for your time. Thank you very much. And the next speaker will be Professor Gabriel Malka. On the question is like tomorrow healthcare perspective. I think that the perspective are starting now that Gabriel will discuss today, and he will propose an overview of the future medicine and the future of medical practices. Thank you, Gabriel. The floor is yours.
As they say, last on the list, but not the least, I will speak now today on the future of medicine and what become the practice of the doctors. Artificial intelligence is among many major topics that are focused on the innovation efforts in the world. Today, we have increased amount of innovative projects associated with bioinformatics, and this field is supported by major technological evolution. So, the AI will transform the method of work. I'd like to share with you on the future of medicine. Our debate today, after defining what is AI, we will see what are some applications, will try to reflect on some issues that concern it. What is artificial intelligence? It's all the techniques or method implemented to build machines that can simulate human intelligence. It is a general concept that covers various realities. AI is emerging from the fundamental research laboratories and is starting to be applied in the world that we are constantly inventing and reinventing the world. In addition, AI has never been so much at the heart of the world debate, and Morocco, our country, is following this trend. Because the AI can foster the social welfare and the human process in according in the United Nations Sustainable Development Goals. We will try to analyze how is helping medicine, doctors, and society in general. What is the application in artificial intelligence in the medical areas? We find many applications in cancer, improved diagnosis and prognosis in sequencing combined with higher resolution imaging. In cardiology, we can find algorithms based on Erwin predict for the pulmonary hypertension and knee prosthesis that detects biomechanical contrast. AI transforms the teacher. The first step, the first level for the AI is the training and the research. AI transforms the teacher's mission and enhances the education system, assuring a success to knowledge and improving scientific research in all fields, as well as the production, management, and sharing of information and knowledge. Each individual will be able to produce a large amount of health data during a lifetime through smartphone or any connected devices. Then the individual is not only source of exponential growth of information that affects the medical fields. Scientific literature and clinical data are exploding to the point that the flow of information is becoming unintelligible to the human brain. You can see that Watson of IBM can read 2 million pages in 3 seconds. And each day we found 3,000 scientific publications in the world, and it's difficult for the human brain to accept all that. For us, use of big data is characterized by 3 things: by the volume, by the variety, by the velocity. There are many challenges around all this information to allow the optimal use. Another application is anesthesia in operating room. In an operating room, artificial operator assists doctors, products of sedative in pre-programmed injection tubes. To practice, to teach for all the students, the simulation center allows the students to reoperate and protocols and to practice. It's mainly about learning to work in teams and to optimize it. can be individually customized some organs. It guarantees a high degree. The high degree of accuracy is for 32 microns, and organ can be customized with 3D printing. The future of medical field is also in nanotechnology. The future of medicine depends on the tiniest details. They are promising, especially in cancerology. And also in home health care. The aging of the population will increase to the number of long-term illness, and the cost is 90 billion euro. A chronic patient treated at home costs less than 200 euro, which is 10 times cheaper than in a hospital. This improves the comfort of life for patients and perform medical survey mission of the lacking area desertification. You can use all the telemedicine, teleconsultation, tele-expertise, and you can also use the companion robot. All the medical fields now can go on with artificial heart to prolong the lifetime, the thought-driven prosthesis, the three-mannequin for practice education, and the question is, will the robot replace the doctors? AI is growing on every level today. Will it change the medical practice, making caregivers and healthcare professionals to reconsider the role and getting patients to be treated in different way? We have to rethink our profession. What reflection, what questions we can use now for use of AI? Health is indeed one of the main fields of application by many companies, and many companies are interested, as Google, Samsung, Apple. We know that Google has spent $6 billion last year only in the health field. Thanks to robots of Watson, it is now possible to sort the useful and the useless, to organize and even follow the evolution of particular pathology. Healthcare designers are about one topic of here, but not just not in the AI. I'm talking about the one that is able to autonomously understand human natural language and imitate human deduction. The assistance for diagnosis by AI. You can identification now malignant melanoma. And Google named the software Show and Tell. And from 130,000 images, the AI learned to make the difference between 2,000 diseases. The detection of micrometastases in lymph nodes and better classification in cancer. On the other hand, some specialties will be transformed, especially those based on signal analysis and as radiology or dermatology. What it happened for the doctors? You find now that the practice of the doctors will be more human medicine. If machine diagnoses the patient and propose the treatment, then what would the doctor do? The doctor is a medical knowledge carrier. The doctor understands what can bring such a tool in the decision making. The machine will not take his place in the decision making process. He will act as an advisor. The doctor becomes as an advisor and a coordinator. His role will be structured and his human competence will be more valued. And what are real impacts of EA? The aim is not to replace the patient-doctor relationship. It's becoming more stronger. From diagnosis and treatment choice making, the AI help to be a guide and assist the doctors. The real issue is not to go faster, but to go further, defining the right objectives. But the risks do exist. Yes, the mistaken risks are not natural. We say that the failure of Google to predict the evolution in the flu pandemic. To avoid these errors, we will have to learn to use these tools and to be aware of their limits and their danger. We should neither be worried nor naive. What is the problem? In one side, we have the good side. The good side with the cure of cancer, we use new anti-infective treatment. We have organ replacement. But in the bad side, we have many problems. Who will have access to discovery, to spend world, what intimacy, all things move. What active place on the robotic world, the place of attending doctor, the risk of type 2 of intelligence. Currently, medicine is still essentially therapeutical, as in the time of Hippocrates. And now, I call this medicine, the new medicine is the 4P medicine. Is a medicine tomorrow is personalized treatment with a prediction, with participative and prevention. Technology is ahead of social issues. Nevertheless, we must ask the question about the reliability of the machines and also about the ethical question to their use simplified medical. Secrecy, transmission of data, limits of the private life, responsibility. Many ethical and legal problems become urgent to solve. The revolution is on the way, and we will not stop it. Are we going to see the replacement of the doctors? The answer is clear. Will artificial intelligence revolutionize medicine? Yes. Medicine without doctor? No. And before I will finish, I will present our program, the new Smart Healthcare City, and I want to tell thanks to two men, Mr. Mustafa Terra, the CEO of OCP Group, and my friend Hisham Al-Habti, the president of the university, to help us to realize a dream, a new model of hospital, a new model of scholarship in medical fields. The application of AI to medicine offers a necessary perspective to the development with new technology. The innovation tends toward one and only objective, to fight death and disease. And I will finish because medicine has come a long way since Turing. We are at the dawn of a new time where AI will take more and more place in healthcare and to help doctors and their work. It is a precious help in the interest of the patients. As François Rabelais said, science without conscience is the death of the soul. Thank you very much.
Thank you very much for this nice topic, Gabriel. It was a great pleasure, and as you know, all of this dream and vision that you have will be reality, and we are working hard to do it, and with you, and until everything is set up, and it's a new and great challenge within the UM6P. And I am honored and glad to be part of that, and it's why we are working so hard to make all of this and all of this new vision in medical practices a new reality. Thank you very much. Appreciate your time, and next event. Donc.